Welcome back to my channel. The tasks will be continued. The chapter molecular basis of inheritance, and today we discuss about replication of DNA, or otherwise we call DNA replication. It is also comes in the exam as a question. Please all see the full video and subscribe my channel for seeing my daily video or everyday video as a crash course. Okay. Replication of DNA. What is the meaning of replication? Replication means DNA make their copy and create their copy by the process of replication. In replication process, DNA make their own copy or make their numbers inside the cell. By the help of DNA replication, it increase the number of DNA. Okay. The replication process is happening by three steps. One is initiation, and the elongation, and last process is termination. In three processes, the replication is be continued. Okay. What is the first basic concept of this replication we study now? The DNA is present in our cell as helical. That means the helical structure of DNA always stable DNA. This helical structure is converted to a linear structure by the help of enzyme topoisomerase. By the help of topoisomerase enzyme, the helical structure converted into straight line structure. Then this straight line structure are disconnected with one strand to another strand. The two strands are disconnected by the help of helicase. DNA helicase enzyme help to disconnect the two sequence of the two strand of the DNA. The two strand of the DNA are separated by the help of the enzyme helicase. Here a case is coming that the two strands are joined after some times, so that SBP single stranded binding protein are using in this case because it cannot be added again. After helicase enzyme, SBP enzyme is uses for it cannot added for that time, so that we use SBP enzyme to synthesize or to separate the two strands permanently. Okay. The upper strand is called as leading strand and the lower strand is called as lagging strand. The upper strand starts from 5' and ends at 3' end, but the lower strand or lagging strand starts from 3' end and ends at 5' end. Okay. Here a enzyme is added that is called as primer. Primer enzyme is coming in this region. Why? What is the work of primer in the replication process? What is the function of primer in the replication process? We use primer when we paint our new door, window. Why we use primer? Because primer try to conserve the window and door so many years, so that we use first primer, then we paint the color. As like this, we first use the primase enzyme, which gives a primer strand. In primer, some RNA basis is present, and after using the primer enzyme or primase, here a continuous process of DNA polymerase is added or is slided. DNA polymerase is slided. Okay. It is happening for the leading strand, and the DNA polymerase added from 3' to 5' direction. That means it's adding oppositely. It is added oppositely, the DNA polymerase strand adding in the leading strand oppositely. First we add the primase enzyme to create a primer. After that the DNA ligase try to make a base before the leading strand, and it is oppositely sliding over the primase. That means 3' direction to 5' direction. In this direction it is slided over the leading strand. But it cannot happening in the case of lagging strand, because lagging strand is a temporary type of primer. We use this RNA polymerase or RNA polymerase primer is called as Okazaki fragment. That means a discontinuous process Okazaki fragment is added. This is called as Okazaki fragment. Okazaki fragment. Okazaki fragment is added to create a base. After that, the DNA polymerase is again used. DNA polymerase here is used, and it is happening continuously. Here the Okazaki fragment is added. Here also the Okazaki fragment is added. After that, in the direction of 5' to 3', the DNA polymerase is used. But after some time, an enzyme is used that is called as DNA exonuclease. Exonuclease enzyme is added in this case. Why? Because the Okazaki fragment is a temporary fragment. Okay, exonuclease help to disconnect the Okazaki fragment or disappear the Okazaki fragment from this lagging strand, so that here a discontinuous manner of the DNA polymerase is created, and after the removal of the Okazaki fragment, the complete DNA sequence is adding in the help of exonuclease. The Okazaki fragment are removed, and in the place of Okazaki fragment, the DNA polymerase again reused or again added. The complete DNA sequence is present. Here also the complete DNA sequence is present, because the primase is a permanent fragment, so that it cannot be removed by the help of exonuclease. After that, DNA ligase enzyme is used to push up or strong the two strand. After that, the two complete DNA sequence is formed by the help of only one DNA sequence. Okay, the DNA replication process is semi-conservative process. Why we called the DNA replication process is semi-conservative process? Because here one primitive strand is present. The both side one primitive strand is present, so that 50% primitive strand are present in the new DNA sequence, so that we call the replication of DNA is semi-conservative process, because the half of the DNA sequence are present in previous DNA sequence are present half of the new DNA sequence. In one DNA sequence we find two DNA sequence. After that, the topoisomerase enzyme again used to help to convert the linear structure to the helical structure. The topoisomerase again used to convert the linear structure into the helical structure. This is all about the process of replication. It is most important. It comes in the exam as a long question also. Please see this video, and many type of short question also comes in this part. It is so important. Thank you all of you listen to see our full video, and like, share, subscribe our channel. Thank you all of you.